Assalamu alaikum and a very good day to Dr. Mahiza. We are from Group 21, would like to present a topic on a control for helminth in horse. As we know, the control is divided into two, which is the first one is non-biological control and the second one is the biological control. Okay, now I would like to explain for the non-biological control, which is the first point is chemical control, which is also known as a drug. For the chemical control, it contains a major classes of anti-helminths. It is divided into three types. For the first type, which is microcyclic lactones, also known as ivermectin or abamectin. This type, it is contain a very broad range of activities, very safe and also very effective at low doses. The next type is tetrahydropyrimidines, or also known as a parental. Okay, for your information, parental it's actually available as a paste or as a tartrate salt that can be used as a daily dewormer mix in the feed. And for your information, parental also it is very effective against large and also small strongyles, ascarids, and also pinworm. Last but not least, for the last type is benzimidazole, or also known as a fembendazole. Okay, for this type, it's actually very effective against most nematode, but not against all those small strongyles or tapeworm. As a conclusion for the chemical control in the part of the non-biological control, every drug differ in every durations of activities. It can be observed by two, which is the first one is egg reappearance period, ERP, and also the developmental stages. Next slide is the example of the parasite life cycle. Okay, as I mentioned above, this is the example of the parasite in horse. The first one is small strongyle, which is known as a Strongyloides vulgaris, tapeworm, the Strongyloides westeri, and last but not least, the pinworm, which is the Oxyuris equi. There's two pictures in the slide. For the right side, it is actually the tapeworm, the Strongyloides westeri, and for the left side pictures is actually for the small
Next,
strongyle.
moving on to the non-chemical control, we can emphasize on the stocking density management. Many horses or many different owners may make it more difficult to control, and it will eventually increase the high stocking density. High stocking density eventually will lead to intensive labor needed to treat each horses individually. So, heavy stocking rates will result in a consistently high level of parasite exposure that even the best deworming program can be no use after all. Moving on to the environmental management. The eggs will hatch and further develop into infective stage larvae with the condition of moderate temperature and moisture. If it's too cold, it will slow the rate of growth. If it's too hot, it will kill the eggs and larvae. So, here to emphasize that by heating the manure, it will eventually kill the parasite, especially during the ascarid eggs. So, we emphasize also on the cleanliness by making sure all feces removed from the pasture to prevent contamination. We can also practice composting for the manure. By composting, it will induce a relatively high internal temperature so the strongyl larvae in manure can be eradicated when the temperature reach over 40 degrees Celsius. And lastly, avoid treatment of equine strongyls during winter in cold temperate climates and during summer months in hot climates. It is to reduce the development of helminthic resistance.
Apart from what was mentioned before, there are other control methods of helminth in horses in terms of management. First one is cross-graze pastures with other species, like allowing horses to graze with goats for example. This is because most parasites are host-specific, which means one particular parasite infects only one particular animal species. So cross-grazing can help to reduce the pasture's burden of equine parasite. For example, goat ingesting equine parasite parasite will halt their life cycle without having any adverse effect on the goat. Second is rotational grazing. This method offers an evasive strategy for controlling worm numbers within livestock. By dividing pastures into sublots of land, horses grazes after the infective stage of larvae have died off. Next is feed hay and grains in raised containers and not on the ground. This is to avoid their feet from being contaminated by feces of the horses that are shedding the oocyte in the feces. Besides, quarantine new horses before introducing them with the existing horses in the farm. This prevents worm from spreading between horses. Also perform fecal examination to ensure that the horses are free from helminth infection. Ensure that the horses are kept in a stress-free environment, such as ensure that the horses' nutrient requirements are met, they have continuous access to clean water, provide shades to prevent heat stress. This is because stress can cause the helminth infection to flare up.
Vaccines. It is injected orally into the mouth of the horses. There are three main vaccines which are applied to the horses, including benzimidazoles, macrocyclic lactones, and tetrahydropyrimidines. Some parasitic stages are inherently less susceptible, so its resistance may be developed, especially when it is applied at the hypobiotic stages, which refers to early stage of the small strong gels. So small strong gels is also called as cyatostomins for the horses, or the adult stages of cyatostomins. We have to deworm the equines by following the schedules. Here are some methods. We can deworm foals every 2 months. We can start deworming foals at 2 months of ages. We can inject ivermectin or moxidectin once or twice a year. And you should know that about individual horses will often require different deworming programs even though they are living at the same place. So we have to slow the development of drug-resistant parasite to reduce the selection pressure for resistance by customizing deworming programs to the farm and individual horses. So here are some examples. We can identify horses most susceptible to parasites and maximize refusal by reducing the overall number of anthelmintic treatments. Refusal refers to any portion of a population that is not exposed to a selection pressure for genetic change. So if the refusal is going to be greater, there will be a greater reservoir of susceptible genes and less resistance in gene pool.
So the next part is the biological control of horse helminth. So fungi has been long known for its ability to infect and kill nematodes. So these fungi are called the nematopathogenic fungi, which can act as a biological control tool. However, control of parasite nowadays is mainly by the use of chemicals, but these commonly used chemicals are losing their effectiveness due to the resistance arising from indiscriminate use. So more people are starting to acknowledge this biological control tool, which are these nematopathogenic fungi. However, these fungi can be divided into three major groups. So the first group is the predaceous fungi. They produce specialized nematode trapping structures on the mycelium. So one of the species are Arthrobotrys species and the Monacrosporium species. And the next group is the endoparasitic fungi. So this type of fungi are obligate parasite of nematodes. They can invade the nematodes by producing the sticky spores which will adhere and penetrate the cuticle of nematodes. And also it can they can invade the nematodes by ingestion of spores by the nematode and lodge into the gut. So one of the species is called Drachmeria coniospora. And the last group is the parasitic fungi. It is known for the ability to attack the nematode at the egg stage. So this is used especially to control animal parasites which have long development and survival time in the egg stage in the environment which is outside the host. So one of the known fungi with this ability is Verticillium chlamydosporium, which attack and destroy the eggs of Parascaris equorum, which is one of the common nematode in horses.